Hello and welcome everyone to Bio-Rad's webinar series on Droplet Digital PCR. This is our 7th webinar in the series Bio-Rad is hosting on Droplet Digital PCR. My name is Mohini Patil and I'm the Global Product Manager for Droplet Digital PCR at Bio-Rad and your host for today's webinar. First, let's discuss the webinar logistics. The webinar will last approximately 1 hour. Dr. Yann Jouvenot will present on ddPCR and its application in environmental studies. Following the presentation, we will have live questions and answers with Yann. Throughout the webinar, feel free to use the Q&A box on the left-hand side of your screen in order to submit your ddPCR questions directly to us. We will get to as many of your questions as we have time for. However, if we cannot get to your question today, we will personally follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. Now, a little bit about our speaker, Dr. Yann Jouvenot. Dr. Jouvenot earned his PhD in human genetics from the University of Paris. Yann completed his postdoctoral studies at Sangamo Biosciences in the Bay Area. Dr. Jouvenot is a staff scientist at Bio-Rad's Digital Biology Center working on the development of various applications. The title of Dr. Jouvenot's talk this morning is Applying Droplet Digital PCR to the Study of Environmental Samples. Without further ado, let's get started. Yann?
Thanks a lot for your introduction, Mohini. As we said, we will discuss today several possible applications of Droplet Digital PCR to environmental studies. First of all, I would like to have a brief introduction on the ddPCR platform, the QX200, for those of you who might be unfamiliar with it, as well as the principles behind digital PCR and its unique characteristics. I'll then describe what is understood as environmental studies, or at least what I understood environmental studies to be. And finally, I'll present a few ways in which digital PCR has been used in these different studies by our current customers and collaborators. This will be illustrated by several publications and their respective results. I'll finally summarize the advantages that are offered by the digital PCR system for this field of work. Here is Bio-Rad's QX200 Droplet Digital PCR system. It consists of two instruments, the droplet generator and the droplet reader. Bio-Rad offers all the necessary reagents to perform digital PCR in droplets. The droplet generator uses a disposable DT8 cartridge to simultaneously partition 8 independent 20 microliter reactions into several thousands of droplets each, all in about 2 minutes. The droplet reader instrument can read a full 96-well plate in about 2.5 hours. This system gives the user great flexibility by allowing for a broad range of sample throughput. By empowering the user to decide how many samples to run, from 1 to several hundred samples can be analyzed in a single workday. At the core, Droplet Digital PCR, or ddPCR for short, is a revolutionary new technology that allows for precise and absolute quantification of nucleic acid. There are essentially 3 simple steps prior to receiving quantitative results. First, the partitioning step, where a standard 20 microliter PCR reaction containing sample, master mix and assay, primers and probe, is divided into about 20,000 uniform nanoliter-sized droplets by the droplet generator instrument. Second, the droplets are transferred onto a 96-well plate and thermocycled to endpoint, usually about 40 cycles. For the third and final step, the thermal cycle plate is placed in the droplet reader instrument, where the droplets are counted individually from each well by fluorescence to provide a positive or negative result. The data is then displayed in the system's software, Quantisoft, as a concentration in copies of target per microliter of reaction. This massive partitioning of a PCR reaction affords a number of powerful technical advantages. First, ddPCR provides an absolute measurement in copies per microliter. One no longer has to extrapolate CQs onto a standard curve in order to obtain a relative quantification. ddPCR will literally count the number of drug molecules present. Second, ddPCR enables sensitive detection of rare events, since partitioning reduces the relative PCR competition between species and allows for a better representation of minority transcripts. Third, because each droplet will give a yes or no answer, ddPCR is much more tolerant of PCR inhibitors than other quantitative methods, and this will be particularly stressed out during this presentation. Finally, the quantification that you obtain by digital PCR is absolute and of high precision, and this can allow to detect subtle changes in either gene expression or copy number variation or even pathogen presence. What is understood as environmental studies? This is an umbrella term that can cover a variety of things, but for the purpose of this presentation, we limited it to the analysis of nucleic acids in samples found in the environment, that is, outside the subject of interest, whether this is a patient, an animal, a plant. This can translate into the detection and quantification of pathogens in the habitat, in food, but it can also apply to the detection of invasive species in habitats or populations. Given that the concepts applied in both types of research, we will also include quantification of genetically modified organisms, or GMOs, as well as some data on the genetic analysis of mosquitoes using ddPCR. First of all, let's go through a summary of the different methods currently used for quantification of DNA in environmental samples. Historically, colorimetric methods have been very widely used, and one of the reasons for that is their great sensitivity. These are methods that start by amplifying the potential pathogens in the sample tested by culturing it in a very rich medium. They can then be plated onto agarose plates or other types of substrates, and using a colorimetric reaction, you can identify the type of pathogen that was initially present in the sample. This affords a great sensitivity to this method, but the number of targets is very limited, as is the throughput. The cost is relatively low, at least in terms of reagents, but not in terms of time and manpower, and of course, it does not provide an absolute quantification. Quantitative PCR, or qPCR, is also very widely used. Its sensitivity is relatively good. The number of targets is limited, but the throughput is a great asset because you can implement several different high throughput formats, such as the 384 well plate, and go through a large number of samples. The cost is low to moderate, depending on the number of replicates that are required per sample. Finally, next-gen sequencing is also making great strides in this field of environmental studies. Although it does not have a great sensitivity in terms of limit of detection, it does provide a wide range of target detection. Of course, multiplexing is the main asset of next-gen sequencing. Its throughput is relatively low, and its cost is, as most of you may know, very high. Finally, how does digital PCR fit in all this? Well, digital PCR provides several main advantages that other technologies do not. First of all, its sensitivity is unparalleled, as has been demonstrated in the previous publications and other webinars that you may have attended. The number of targets is also an asset, since we can readily analyze 4 different targets at once. The throughput is very good, since it can use 96 well targets, and for each well or each sample can provide a statistical confidence interval. The cost is relatively moderate, especially taking into account the reduced number of replicates that one needs. And finally, it is the only method that really provides absolute quantification of the nucleic acids in the samples. This robust method has also demonstrated a reduced sensitivity to PCR inhibitors, and this is a great asset to the detection of rare targets in complex matrices, especially as the ones that you can find in environmental samples. So I'd like to take a pause here and ask the audience to fill in this poll and just tell me what methods do you currently use for nucleic acid detection in environmental samples. This will take just a few seconds, and we will just linger on this slide until we collect a few results and move on to the rest. This is purely informational, and in order to see what type of tools are being used currently in the field. So we have either, of course, colorimetric methods, which can include a wide variety of different medias and substrates, qPCR or even QRT-PCR, next-gen sequencing, digital PCR, or any other method that you might see fit. Okay, another few seconds, and let's see what kind of results we're obtaining. So it sounds like we have a very clear winner. qPCR is definitely the most popular method used right now, which sort of correlates with what the publications have been showing. About 63% of you are currently using qPCR for environmental studies. Digital PCR is making its way over there with 26%. And colorimetric methods seem to be the last choice for this audience, at least. Thank you very much for your time. Moving on to customer results, we'll go through several publications that have used digital PCR for environmental studies. The first one was published by Rothrock and collaborators and dealt with the quantification of bacterial pathogens in commercial poultry processing plants. So in this study, Dr. Rothrock and his collaborators used digital PCR in comparison with qPCR to detect several different types of bacterial DNA in the different samples that are generated from poultry processing plants, specifically water samples. In this slide, you can see the comparison between qPCR and ddPCR from quantifications of pathogens of bacterial ribosomal DNA in samples from the scalder water or samples from the chiller water. These are two different tanks that are used for processing the poultry carcasses. And it is very important to monitor the presence of pathogens in each. As you may see, these two different graphs show similar sensitivity, or even in the case of the chiller water, a little bit higher, between digital PCR and qPCR. But what is specifically striking is on the samples from the scalder water, the much more robust and precise quantification of this method. The authors move on to do another type of study detecting specific pathogens, Salmonella, C. jejuni, and Listeria, in samples from the scalder water, but at different points in the process. At the early start, then in the middle, and finally at the end of the process. These pathogens are detected either by culture methods, digital PCR, or qPCR. And what can be seen here is that in several cases, at least for C. jejuni and Listeria, digital PCR detects the pathogens with greater sensitivity than qPCR, and of course the culture methods. The conclusions from the authors were that given the much higher throughput and absolute quantification of ddPCR, which produced statistically similar results to qPCR in this study, this technology represents a significant improvement in the molecular detection and quantification of zoonotic pathogens in commercial industry environments. The next paper was published by Dr. Rachi and collaborators and used digital PCR to detect and quantify waterborne RNA viruses in different types of water samples. In this study, Dr. Rachi and his team took water samples from different rivers, streams, and lakes and tested them for all the different environmental parameters, pH, color, conductivity, turbidity, and analyze them in parallel by RT-ddPCR or RT-qPCR. They spiked all of these samples with similar amounts of rotavirus RNA. And what can be seen on this table is that, on average, ddPCR detects higher copy numbers than qPCR, which is reflective of a higher sensitivity. But, more to the point, it has a greater precision, as can be reflected by the lower CVs. They also demonstrated, by spiking the rotavirus RNA into the water samples, that ddPCR had a lower limit of detection in this type of matrix. They attributed this to the increased tolerance to PCR inhibitors that was already proposed earlier. Another publication from the same team of Dr. Morissette et al. Uses digital PCR to quantify GMOs in food and feed samples. And the rationale for this was that since GMOs are becoming more and more prevalent in modern agriculture, it is necessary to develop more sensitive methods to detect and quantify them. The problem is that the current qPCR methods have a high uncertainty levels, and they can also be inhibited in some complex matrices. So they developed a duplex assay for both transgene and crop endogenes using digital PCR. The results are summarized in these tables. What you can see here is that digital PCR ensured the precise measurement of GMO content across a wide range that corresponded basically to the routine laboratory range, from 0.1% to 30%. The bias, or the value in brackets, is basically the divergence from the theoretical or expected value, the target value. As you can see, by comparison with the qPCR results, the level of bias of digital PCR is lower. Not only that, but in a few samples, in 5 samples, qPCR did not detect the GMO, whereas digital PCR did detect it. Dr. Morissette, pardon me, I clicked a little bit too fast here. Dr. Morissette and his collaborators also did a cost analysis for these methods and compare them, basing their comparison in statistically comparable data. They evaluated that they needed 4 plates to test about 96 reactions. And they compare the cost and the time needed, especially the hands-on time, between the two methods. And it turned out that digital PCR resulted both in a reduced cost in time and reagents. In this study, the researchers discovered a new Y-specific gene in the Asian malaria mosquito by NGS. Digital PCR was used to determine the copy number of this gene, and they were looking for a Y-specific gene that was possibly expressed earlier in development and that could trigger sex determination. This gene, GUY1 or GUY1, was therefore kind of the sex determination gene in mosquito, and they needed a straightforward method to quantify the copy number in their samples. Albeit this is not an environmental study per se, in the fact that the sample itself is not environmental. This is extracted mosquito DNA. It was included in this presentation given the nature of its model. Mosquitoes are very prevalent in many different public health and environmental studies. What was found in this article was that digital PCR provided excellent interpretation of the data. As you can see here, the 3 male samples all came out at a copy number of 1 for the GUY1 gene, and the female samples came out at nothing. Digital PCR, therefore, was determined to be a useful method for study of mosquitoes and their genetics. These results are not yet published, but should be soon. And they deal with the use of digital PCR to detect the spread of invasive species. In this case, the Asian carp. Asian carp is an invasive species in North America. It was introduced many decades ago by sports fishermen and started migrating out of its initial ponds. Several ecosystems have been endangered by the carp. The Army Corps of Engineers in 2012 was actually tasked to stop the spread of the carp. But in order to assess the efficacy of any control method, it was necessary to have a sensitive and quantitative technique to determine the carp population. Scientists at the DBC helped a team of scientists at Cornell University, under Jim Casey's supervision, to develop a method to quantify mitochondrial carp DNA in lake water. This method is currently being tested to be used and to assess the spread of Asian carp, specifically the spread towards the Great Lakes, where the Army Corps of Engineers is currently developing a set of barriers to stop the invasion. This is just a brief demonstration of the 2D plots between the environmental samples and the control sample for the big head carp. We get a perfect alignment of the different clusters in an orthogonal fashion. Oops, pardon me. I went backwards a little bit. In this case, we tested the level of PCR inhibition from the different lake samples, and this was just testing the internal positive control. As you can see, the value for the internal positive control is not affected at all by the different types of samples studied. This was then used to quantify the amount of carp mitochondrial DNA in these different samples, and as you can see, they vary widely. And they were later summarized and compared to their qPCR results in this table. The outcome of this was that digital PCR provided a much more sensitive and much more robust quantification of the mitochondrial DNA. They also saw that in some samples, for instance in the Lake Chicot sample, no amplification was actually obtained, not even as a positive control, but digital PCR did give a value. All this data showed that digital PCR was definitely a method of choice for a detection of invasive species, especially in large water bodies. Finally, to summarize, what are the benefits of utilizing digital PCR for environmental studies? As I mentioned before, ddPCR is a sensitive and precise method, and it provides an absolute quantification of RNA and DNA molecules. It does provide a better reproducibility for quantification of low abundance targets, and this is particularly useful in cases where you are detecting rare pathogens or rare species in the medium. It also provides a reduced sensitivity to PCR inhibition, which allows for the study of difficult matrices. A team of scientists actually used this in many different, with many different known PCR inhibitors, and determined that on average digital PCR resisted to higher levels of these inhibitors than qPCR. The QX200 system also has a much higher throughput and lower cost per sample in the digital PCR market, which makes it the best option for a systematic testing of large numbers of samples. Finally, it is the method of choice, or at least should be the method of choice, for several environmental uses of PCR, including pathogen detection in food or habitat, or detection of invasive species, or finally even assessment of water quality in either seawater or fresh water. I'd like to acknowledge a few of our collaborators that helped with the results. And finally, these are just a few of the publications that were reviewed here. If you have any questions regarding this or any others, I'll be pleased to answer them. Thank you very much for your attention. I'll be now taking your questions.
Yann, thank you very much for that very engaging talk. If the audience would like a copy of today's PowerPoint presentation, a copy of the QX200 ddPCR brochure and ddPCR applications guide, they are available at the link to the left of your screen for download. The webinar is also available on demand within 24 hours. If you would like any additional information, please feel free to reach out to your local Bio-Rad ddPCR specialist as well. As a reminder, please feel free to use the Q&A box on the left-hand side of your screen in order to submit your ddPCR questions to us today. Yann will now take questions. Our first question is, Yann, how does the QX200 compare to other digital platforms for environmental studies?
Well, I can't say really I have seen data from other digital platforms out there. What I do know, though, is, at least based on the specifications, the QX200 does provide the ideal combination of throughput and dynamic range. So some of the current platforms that are being commercialized do provide an equivalent throughput on a 96 well format, but they have a very, very limited dynamic range, which limits the number of users that you can do with this technology. Others can have an equivalent dynamic range, but then have a much lower throughput, or actually a much slower throughput as well. So all in all, I believe that the QX200 does provide the ideal package for these type of studies.
Do you see the ddPCR technology being applied in sanitation systems or in soil-based site monitoring?
So we don't have any feedback from sanitation systems, really, but I cannot see a reason why it would not be. This is definitely something that many different teams in pathogen detection, pathogen safety labs have been exploring. So I believe that in the coming years we will see a lot more of the digital PCR uses in water sanitation plants and other types of facilities as such.
What is your opinion on the importance of RNA DNA extraction and purification for accurate quantification?
That's a great question. In as much as it is possible, I believe that the sample, if it can be purified, should be purified. But if this does not match with the workflow, or if by any chance the targets detected in the sample would be lost by the precise purification test, then this can be foregone. We have seen qualitative differences, but not necessarily quantitative differences in the levels of purification between different samples. So what I mean by qualitative differences is that you can basically observe differences in the 2D plot or 1D plots, maybe an increase in intermediate droplets in unpurified samples. And this is reflective of a certain level of inhibition. But what we have seen also is that once the PCR is actually at a level where you can obtain a cluster, a well-formed cluster, this affects very little the final quantification of your target. So I don't know if I gave a very clear answer to that question. Whenever possible, of course, digital PCR will work better on purified DNA or RNA. But when it is not possible or not recommended, then it will definitely give much better quantification than qPCR.
So along those lines, our next question is reagents related. Can master mixes from other manufacturers be used with the QX200 platform?
No, and the reason for that is that the chemistry of the QX200 platform has been really optimized for the use with its current master mixes. What could be observed if somebody were to use a different master mix would be, you could interfere with the chemistry between the oil and the mix and the aqueous phase, and therefore not form droplets, or otherwise form droplets that are of different sizes or different uniformity in sizes than the ones the software or the instrument should be detecting. This will actually alter your quantification of the target, and in a certain way also will probably affect the efficiency of your experiment. So while Bio-Rad is developing more and more master mixes to suit the needs of our customers, we definitely cannot recommend the use of anything else than our current ones. We understand that there are some specific master mixes that some customers need to see, and actually we really value the input for those. So if your research requires a specific type of master mix, please contact Bio-Rad and let us know what that type is, and we will make sure to incorporate that knowledge in our pipeline of development.
The next question is regarding contaminants. Are there any known contaminants from samples that are incompatible with droplet formation or stability?
Yes, there are known contaminants for which we can do much about. So the first and obvious ones would be detergents. So detergents, for instance, would affect just the plain formation of the droplet, the micellation. So if the sample you are testing contains detergents, at least high concentrations of detergents, because I should specify we can work with low concentrations of certain detergents. We have done some of those tests, but and that works fine. But if you go to much higher concentrations, it will prevent the formation of the droplet itself. Other known inhibitors that will definitely affect your results, yes. I would say anything that directly attacks or degrades the activity of the polymerase will affect also digital PCR. If you have, for instance, a phenol contamination or any type of chaotropic agent maybe that might denature the protein, that will definitely interfere with digital PCR results.
Our next question is assay related. Does Bio-Rad have assays for water pathogens, or do we provide primers and probes for certain pathogens?
We currently do not provide specific assays for water pathogens. That is definitely something that is being explored, though, since there is that need out there. What I would recommend is contact your local rep and make them aware of that question or that need, and they will definitely relay it to the DBC, and we can potentially provide you a solution. So we value all that kind of input.
Well, Yann, that was our last question for today. Thank you, everyone, for attending the Bio-Rad Droplet Digital PCR webinar. As a reminder, today's webinar will also be available on demand within the next 24 hours. For those of you who had questions we were not able to get to, someone from Bio-Rad will follow up with you over the next few weeks. For more information on Droplet Digital PCR and educational resources, visit www.biorad.com. Thank you and have a great rest of the day.